Hello, everyone. I'm Jeff Lord, and this is our last session of the regular Rare Fair for this conference. And this year we decided to do something a little different in the afternoon. We wanted to feature some technology that was happening in the rare disease space. So Homology Medicine, one of our sponsors, has provided us with a video, and they're going to talk about some of their gene therapy. And you're going to be hearing from Kara Mayfield from Homology. And so I'm going to share my screen and play that video. And a special thank you to Homology for participating in our Rare Tech Showcase.
Hi, everyone. Thank you so much for the opportunity to speak with you today about Homology Medicine's genetic medicines technology. My name is Kara Mayfield. I'm the Vice President of Patient Advocacy and Corporate Communications at Homology. What I plan to talk about today is listed here. First, I'll discuss Homology's technology platform, and then I'll talk about how we've advanced that platform into gene therapy and gene editing candidates. I'll end with information for us to be able to get and remain connected with any questions, comments, or feedback you might have for me. So since day one, Homology is focused on our mission to cure genetic disease. We're at a point in time where we have the technology and understanding of disease biology to potentially cure genetic disease. And here is a snapshot of who Homology is as a company. If you start in the middle of the slide where it says technology, our company was based on the discovery of a set of viral vectors called AAVs that are naturally derived. In fact, they were found in human stem cells, so we call them AAV HSCs. And we believe the fact that they are naturally derived may translate into having a better immune profile in patients. AAV HSCs are delivery vehicles for genetic material. We have characterized their broad biodistribution properties, which is basically where the vectors would go in the body and what organs and tissues they would target. And what we found is that they can all target different tissues, which is important for the ability to treat different diseases. Most AAVs go to the liver, some better than others, but they can also go to skeletal muscle, cardiac tissue, and other organs within the body. What's unique about the AAV HSCs is that they have the ability to cross the blood-brain barrier following a single IV infusion. So that's also important for diseases that have a neurocognitive effect. So we have the technology and we've coupled that with the people. So our team has been focused in rare disease drug development and have brought forward 11 rare disease drugs throughout their careers. We also have the ability to manufacture our treatments. We actually have a facility right next to our headquarters in Massachusetts. And when you combine these things, you'll see we've been able to develop 5 genetic medicines that are either in preclinical development or are already in clinical trials. Our 3 clinical trials include a phase 2 study with our HMI 102 gene therapy for phenylketonuria, or PKU. Our phase 1 study with our HMI 103 gene editing candidate for PKU. And then we also have a phase 1 study with our HMI 203 gene therapy candidate for Hunter syndrome. And I will talk about those more in just a minute. But before I go into more detail about our technology, I want to just set the stage for one of the types of genetic diseases that our technology can tackle. As you likely know, many genetic diseases are caused by mutations or problems within a single gene. Our DNA makes up our genes, and they act as instructions to make proteins that are necessary for the body to function normally. In a genetic disease, a gene is either missing or mutated, which means it lacks the instructions to make a working protein. In an autosomal recessive disorder, which are the most common types, and they occur when a person inherits 2 mutated copies of a gene, one from their mother and one from their father. Some examples of these include PKU, like I mentioned, cystic fibrosis, sickle cell disease, metachromatic leukodystrophy, and others. There are also autosomal recessive diseases that affect males primarily, as they're linked to the X chromosome. So here are the three ways in which we are applying our AAV-HSC technology to address genetic disease. We're doing gene therapy, GTX-MAB, and nuclease-free gene editing. And so what's important to note is that all of these approaches use our AAV-HSCs as the delivery vehicle, and all are designed as a one-time treatment. As I just described, because we have characterized our AAV-HSCs so much, we know which ones target the particular area of the body that is needed for a certain disease. And we can then also choose which genetic medicines approach would work best for a particular disease. So now I'll go through those three arms of our platform, one by one. So starting with gene therapy. Gene therapy involves introducing a functional copy of a gene into a patient's cells where there's a missing or mutated gene. The functional gene exists inside the cell with a set of instructions to create the needed protein. So in this example, the AAV-HSC is packaged with a functional gene and a promoter, which is used to turn on the gene once it reaches the cell. When it gets inside the cell, the gene and the promoter create a circular episome, which you can see there in the cell depicted in blue. And then that works to create functional mRNA that writes the instructions for the body to create a protein or an enzyme. You can kind of call it either way, and is represented by the blue image in the outer part of the cell. We are using this approach in our HMI-102 gene therapy program for adults with the rare disease PKU. So, as I mentioned before, PKU is an autosomal recessive disease. It's a rare inborn error of metabolism, and it's caused by mutations in the PAH gene. So people with PKU are lacking the functional enzyme needed to metabolize Phe from dietary protein. If left untreated, PKU can result in progressive and severe neurological impairment. And currently, there are no treatment options for PKU that target the underlying genetic cause of the disease, like we're trying to do with HMI-102. So our gene therapy is being evaluated in a clinical trial in adults with PKU. And that clinical trial is called Phoenix. So to the right, you can see how HMI-102 might work for a patient, or at least how it is designed to work for a patient. Patient would receive a one-time IV administration of the gene therapy. The gene therapy is designed to target the cells in the liver. Once it enters the cells, it's designed to deliver the functional gene with the promoter, like we just talked about. And then the functional gene would work to create the functioning protein or enzyme that is missing in somebody with PKU. The clinical trial with HMI-102 is ongoing. Like I mentioned, it's in phase 2, and we plan to report initial data from that trial in mid-2022. We are also using a gene therapy approach in our program for Hunter syndrome. Now, Hunter syndrome is another autosomal recessive disorder, and it's a lysosomal storage disorder caused by mutations in the IDS gene. It leads to toxic lysosomal accumulation of GAGs, which are large sugar molecules or cellular waste. Severe Hunter syndrome causes progressive debilitation and intellectual decline, and there are no treatments currently available to address both the cognitive and the peripheral organ manifestations of this disease. So we are evaluating our candidate HMI-203 in a clinical trial called Jumpstart. And to the right, you can see how it might work for a patient, which is very similar to what I described for our PKU trial, except that this therapy is designed to provide copies of the IDS gene to target more organs and has the ability to cross the blood-brain barrier. Now moving on to the next part of our technology platform, which is gene editing. So gene editing replaces the gene that is causing a disease, which is different than what I just described for gene therapy, which provides the functional copy of the gene, but it doesn't replace the missing or mutated gene. So we use what's called nuclease-free gene editing, which means our approach does not cause any cuts to the DNA, like you might have heard of with CRISPR or other approaches. It actually uses the body's natural DNA repair process called homologous recombination. So we've always known that AAVs have the ability to induce homologous recombination, but previous work with other vectors has shown it happening at a very low rate, so not enough to be suited for a therapeutic development. But then when we discovered these AAV HSCs, we realized that they could do it at much higher rates, and therefore they could be good therapeutic development candidates. So to understand our approach to gene editing in a little bit more detail, if you look at the image, a working copy of the gene is packaged inside the AAV HSC alongside what are called homology arms. Now, these are long stretches of DNA that help the gene find the exact location in a person's DNA to replace it. We've learned a lot over the years of working with these AAV HSCs about how exactly to design the homology arms, including different lengths on either side, depending on the areas that we're targeting, and also what areas of the genome are most amenable to editing. So if you go back to the picture, once inside the cell, the gene with the homology arm finds and replaces the gene that is causing the disease. Then it works to create the mRNA, which write the instructions for the body to create the enzyme or protein. So for gene editing, it's critical to evaluate on and off target integration or editing, which means whether or not the gene integrates in the right location or if something else is integrated that is unintended. At Homology, we've developed assays to assess this, including genome-wide analysis. And because of the homology arms, we can look throughout the entire genome to see if it's integrated anywhere but where it should. Other technologies can use predictive algorithms to assess this, but we take this genome-wide approach with next-gen sequencing, and we haven't found any unintended integrations in our preclinical models. So we are using this approach for our HMI 103 gene editing program for pediatric PKU. Now, the reason we have two approaches to PKU relate to cell division. So both our gene therapy and our gene editing approach to PKU target the liver, which is where the enzyme is made. An adult's liver is not rapidly dividing, so providing a copy of the gene to the cell could have a lasting effect. Now, a child's liver is still rapidly dividing, and the cells are still, rapidly growing, the cells are dividing. So to provide a lasting treatment or potentially a cure, a child would need a permanent DNA correction, or else the treatment would lose effect as the cells divide, and then the treatment would get diluted. So this approach, our HMI 103 candidate, is being evaluated in a clinical trial. To the right, you can see how it might work for a patient, which again is similar to our other trials, except we are delivering a gene editing product instead of a gene therapy. And lastly, we have our GTX MAB part of our platform. Our GTX MAB platform is an extension of our gene therapy approach. It is designed to leverage our AAV HSCs to deliver a gene therapy using a construct with therapeutic antibody proteins that are delivered to the liver. And there they would form fully functional, full-length antibodies and are secreted throughout the body. So this is earlier in development, but it does represent the opportunity to help even larger patient populations. In fact, our first program is not targeting an inherited genetic disease, but rather an acquired genetic disease. So our development candidate, HMI 104, is targeting PNH, which is a rare, acquired, life-threatening blood disease caused by mutations in a single gene, and it causes red blood cell destruction. We chose this area because it is a well-established safety profile with current therapies, current antibody therapies, but there's still a large unmet need that could potentially be addressed by a one-time treatment. We're in preclinical development, but you could see on the right how it could potentially be used in humans. So again, you would start with this IV injection to express the antibody. The liver secretes the antibody, which then gets circulated into the body. And in essence, with a one-time treatment, you would enable the liver to become a factory that continuously produces the antibodies versus current therapies for this disorder, which would have to be administered chronically and could be associated with the peaks and troughs that patients can experience and can be problematic. So I know that I've spent a lot of time here talking a lot about our science, our technology, drawings of treatments and cells, but I do want you to know that Homology is made up of a team of motivated and dedicated people. And our job in patient advocacy is to understand the needs of the communities we serve and to bring that awareness and feedback back to our teams. And our teams through, from R&D all the way through manufacturing and commercial, are motivated and inspired to learn more and support the patient communities beyond their day jobs of bringing new treatments forward. So with that, I just want to thank you again for the time and the opportunity to speak with you. Because we're not live and I don't have the opportunity to meet you guys in person right now, I just wanted to leave behind 2 ways that you can get in touch with Homology. If you have any questions about what I just presented, you can email the patient advocacy team at patientadvocacy@homologymedicines.com, or you can always reach out to me at cmayfield@homologymedicines.com. I'd be happy to hear from you. So with that, thank you and enjoy the rest of the conference.
Well, thank you, Kara, for sharing about the technology that Homology is using. It's just fascinating. We talked about the gene therapy process, and thank you so much. We will be hosting Rare Tech showcases each month. So our next one will be in February, and we're hosting one each month following that. Thank you all for your time today in attending. We hope you enjoyed the presentation. Thanks to Homology again for providing us with some Rare Tech showcase material. Enjoy the rest of the conference, and don't forget to come back for the replays.